So we have discussed how the leaves, if the margins of the leaf they touch the midrib, they produce small leaflets, giving rise to compound leaves. And we have discussed about venation, which is arrangement of the leaves, sorry, arrangement of veins on the leaf. It can be either reticulate or parallel. Parallel venation is mostly seen in dicots, and reticulate venation in, sorry, parallel venation is seen in monocots. I'm sorry, parallel venation is seen in monocots, and reticulate venation is seen in dicots. Now let's understand what are the main functions of a leaf. We know that the main function of a leaf is to perform photosynthesis. Okay, synthesis means to produce something, photo means using light. Okay, so basically, leaves perform photosynthesis. What is photosynthesis? It's the process where leaves utilize the water that we provide. They take in carbon dioxide in the presence of sunlight, and using chlorophyll they convert this carbon dioxide and water into glucose, which is a sugar, and they give out oxygen and energy. Is it clear? So leaves take in carbon dioxide with the help of structures known as stomata. Stomata help in exchange of gases, right? They take in this carbon dioxide using stomata, and water is taken up by the plants through the roots, and sunlight falls on the leaf blade. Okay, chlorophyll they are present inside the leaves, and this the plant converts it into glucose, a type of sugar, and it gives out oxygen. Okay, so during the process of photosynthesis, what actually is happening? Plants take in carbon dioxide and give out oxygen. And during this process, a sugar is being synthesized, which is known as glucose. Okay. Now, for example, let's assume there is a plant which is kept in sunlight. Okay. And it was a very hot day from morning till evening. There was a sufficient amount of sunlight. So there was continuous process of photosynthesis happening inside the plant. So what's happening? Plant is taking in carbon dioxide, utilizing the water which is present in the soil, and converting it into glucose, the sugar. Okay. Now excess glucose, whatever is synthesized by the plant, it gets stored. Excess glucose is stored in the form of starch. Like for example, let's assume your mom made sweets for you. Okay, she made your favorite laddus. Like for example, she made, for example, 50 to 60 laddus. You came back from school and you were surprised that your mom has made your favorite dish. Okay, now she'll offer you one or two. Okay, one or two laddus. What happens to the remaining amount of laddus, remaining 48 laddus or 58 laddus? Will you eat the laddus on the same day? Obviously, no, right? She'll take a box, store the laddus in the box in an airtight container. Yes or no? In the same way, here the plant is preparing its food, and that is glucose. Now, all the glucose that it is synthesizing, it won't utilize it. It stores some amount of glucose in the form of starch. Is it clear? So basically, during the process of photosynthesis, where light is required, sunlight is essential for photosynthesis, and chlorophyll pigment is also needed. So what do plants do? They take in carbon dioxide, utilizing the water present in the soil, convert the light energy into chemical energy, which is sugar. Sugar is being produced. What is that sugar? Glucose. And excess glucose is stored in the form of starch in the leaves. Clear? And which structure is helping in this exchange of gases? The leaves only, because leaves only have what in them? Stomata. Okay, so stomata basically is the structure that is involved in the exchange of gases. And where is the stomata present? On the lower surface of the leaf. Okay, the lower surface of the leaf has the stomata. And leaves also help in transpiration. What is transpiration? Now try to understand. Whatever water that is provided in the soil, the leaves absorb that water. Not all water is utilized by the plant. Excess amount of water is lost. Okay, that is called transpiration. Excess amount of water that is present in the plant that is lost into the atmosphere is said is called as transpiration. And how is that water loss happening? With the help of stomata only. Is it clear? So what is transpiration? Usually, this water that is provided in the soil, plants absorb through the roots. Now, not all water that is absorbed by the plant is utilized for its needs. There is some excess water. If it absorbs it, if the plant absorbs any excess amount of water, it basically tries to see that the water must be eliminated. Now, this elimination of excess water is only known as transpiration. Okay, here you can see a picture of a scientist. Basically, is Jan Ingenhouse. Now, what is the contribution given by Jan Ingenhouse? He said that plants during the day, when there is sunlight during the day, plants they take in carbon dioxide and give out oxygen. And this process only we are calling it as photosynthesis, right? And he said during the dark or night time, when there is no sunlight, plants they take in oxygen and give out carbon dioxide, and this process is called respiration. So what is the contribution of Jan Ingenhouse? He said that plants perform photosynthesis during the day where they take in carbon dioxide and give out oxygen, whereas during the night, in the dark, respiration is performed where plants take in oxygen and give out carbon dioxide. This occurs during respiration. Whereas this process where carbon dioxide is taken in and oxygen is given out, it occurs during photosynthesis. Clear? So that was the contribution by Jan Ingenhouse. So these are the basic functions of a leaf. A leaf helps in performing photosynthesis. It helps in exchange of gases through stomata and also helps in uh, elimination of excess water absorbed by the plant through transpiration using the stomata structure. Okay, and this is the equation of the photosynthesis where carbon dioxide combines with water in the presence of sunlight and chlorophyll to give glucose, oxygen, and energy. And excess glucose is stored in the form of another type of sugar called as starch. And here is the picture of the scientist Jan Ingenhouse, who contributed by saying that plants during the dark they perform respiration by taking in oxygen and giving out carbon dioxide, while during the day in the presence of sunlight, they take in carbon dioxide, give out oxygen, and perform the process of photosynthesis where they synthesize sugars like glucose. Clear? Now, here is an experiment to show that starch is present in the leaves. I told you leaves they perform photosynthesis where they absorb sunlight and give glucose. This excess glucose is stored in as starch. To prove that starch is present in the leaves, you can do an experiment. Okay, take a variegated leaf. Okay, what is variegated leaf? A leaf which has white patches along with green patches. Okay, something like this. Here you can see a variegated leaf. Okay, now this variegated leaf, first put it in boiling water. Okay, after putting it in boiling water, transfer the leaf into a bowl of alcohol that is boiling. Here it is boiling water into which you have transferred the leaf. Then transfer the leaf into a bowl containing boiling alcohol. Okay, now what does alcohol do? It completely sees that the color is removed in the leaf. Leaves are usually green in color. Why? Because of the presence of chlorophyll in them, right? Here you can see that it has here the leaf. Here it is white in portion, whitish in portion. Here the leaf. This portion of the leaf is green in color because of presence of chlorophyll. But when you put that leaf in the boiling alcohol color is actually transparent. Okay, but after boiling and putting the piece of leaf, the alcohol becomes greenish in color because it takes all the color of the leaf and leaf becomes transparent. Now what you do is add iodine to the leaf. Okay, Your leaf, initially it was green in color here and it is white, okay, to the other side. Now when you place it in alcohol, complete leaf is transparent, okay. Now add few drops of iodine to the entire leaf. Wait for 5 minutes and observe the leaf. What happens is the portion where the leaf is green in color, that portion of the leaf, you can see blue black color upon addition of iodine. Why is that so? Try to understand. Let's assume this is your leaf where here it's green in color. Let's assume because of presence of chlorophyll, and here this is the whitish portion. That's why we have picked a variegated leaf. Okay, now you put it in water first and then in alcohol. When you put it in alcohol, what happens? Complete leaf gets decolorized. Okay, no color is seen. Now add few drops of iodine. What happens when you add iodine? We know that this is green portion of the leaf. Can the green portion of the leaf perform photosynthesis? Yes. So this portion of the leaf can take in carbon dioxide, utilize the water, can perform photosynthesis and synthesize glucose. Here, glucose molecules will be present. Excess glucose gets stored into what? Starch. So, here, starch can be seen. Now, upon addition of iodine to this region, what actually happens? Starch reacts with iodine to form blue black color. This picture, try to concentrate on this picture. You can see a variegated leaf where here it is greenish and the outer side it is whitish. When you pour iodine, what actually, after doing the steps, that is put the leaf in boiling water, then put it in alcohol to completely decolorize it, then add a drop of iodine, you can see blue black color is not seen on the entire leaf, it's seen only in the green portion. Why? Because only the green portion performs photosynthesis, only the green portion synthesizes glucose, only in the green portion the excess glucose gets stored in as starch. So the green portion has starch in it. So the starch which is present, it reacts with iodine to give you the blue black color. Okay, so this experiment can be done in your lab to prove that actually during the process of photosynthesis, glucose is synthesized where excess glucose gets stored in the form of starch. Clear? And for this, it's better you take variegated leaf to understand that the white portions cannot perform photosynthesis. If you take a leaf which is entirely green, then entire leaf shows you blue-black color because the entire leaf blade can perform photosynthesis, right? So this is a simple experiment that can be done in lab to prove that leaves perform photosynthesis, and during this process of photosynthesis glucose is synthesized where excess glucose is stored as starch. Okay, so we have discussed the basic functions of a leaf, where leaves play a very important role in performing photosynthesis, in transpiration, and even in exchange of gases. Now let's understand modifications of leaf. Okay, what are the various modifications in a leaf? First is leaf tendrils. In some plants, the stems are very weak. For example, here you have pea plant and glory lily. Okay, this is glory lily plant and this is pea plant. Here the stem is quite weak. Okay, so that's the reason leaf apex. We know the leaf apex, right? This is the apex. The leaf apex modifies into tendrils, and these tendrils, if you see here, the leaf apex is modified. It's getting modified into tendril. So what do these tendrils do? They take support from other plants and help the plants to grow, to climb. Okay, so this usually the stem provides support to the plant, but here the stems are quite thin, they are weak. So here the in order to provide support to the plant, the leaves get modified into tendrils. These tendrils twine around strong support. They twine around the other plants and help the main plant to grow. Clear? So tendrils are basically modifications of tendrils can be modifications of either stem or leaf. Okay. In my previous video, I explained about stem tendrils, where stem gets modified into tendrils in some plants. But here we are talking about leaf tendrils where the leaf is getting actually modified into tendril. Okay, if you remember stem tendrils, we discussed in grapevine, guard plant, passion flower. Okay, there you can see tendrils are modifications of the axillary buds, sorry. But here the tendrils are modifications of the leaf. But in both cases, whether it is in grapevine plant where the tendrils are modifications of the axillary bud, or pea plant where the tendrils are modifications of the leaf, in both cases, the tendrils play a key role in providing support to the plant. Okay, since the main stem is weak and thin. Next is scaly leaves. If you remember, these are your bulbs. What are bulbs? Underground stems, right? If you see, the stem is bulb, which is a modification. Bulb is modification of stem. Here you can see that the stem is surrounded by a terminal bud, and these terminal buds are surrounded, terminal and axillary buds are surrounded by leaves. If you see in an onion, these are all leaves. These leaves are called fleshy leaves. But outer peel, can you see, which is completely dry, where which your mom actually peels it off, right? The peel that you peel it off, that is they are called scaly leaves. So scaly leaves are dry and fleshy leaves they store food. Okay, so the leaves are modified to store food in them. Here, where fleshy leaves they store food, scaly leaves are for protection of the bulb, to protect the bulb. Okay, you can see this in onion and even in garlic. Now coming to ginger. Ginger, what is the stem called? Rhizome, right? Underground stem is called rhizome. And here you can see these are your nodes. Okay, and this region is called internode, right? You can see nodes here. And from the nodes, can you see these brown color dry structures? They are basically your leaves that are arising. Okay, it's a leaf. Even this is a leaf. The brown structures that you see. And these are dry scaly leaves. Okay, and they are very small in structure. So these are the leaves, that scaly leaves that are, like here you can see modifications of the leaf, which mainly perform the function of storing food. Examples include ginger, onion, garlic. In onion and garlic, you have fleshy leaves as well surrounding the terminal and the axillary bud, helping in storing the food, and the scaly leaves act as protection to the bulb. Okay, and in ginger, the leaves, the scaly leaves, are quite dry and they are very thin. Okay, next is spines. Spines are also modifications of the leaves. Thorns are different. Okay, thorns, if you see in bougainvillea, we have seen thorns, right? They are modifications of stem, modification of the axillary bud into pointed structure. So try to understand these are modifications of stem, but spines are modifications of leaf. What are spines? If you see opuntia, we talked, I gave you example of opuntia even in stem modifications. Why? Because the structure that you see, like this green color structure, it is not leaf, it is the stem, which is known as phylloclade, or it is also known as cladode, right? They are modifications of the stem. And can you see these pointed structures? They are called like thorny structures. They are not thorns, but thorn-like structures. They are called spines. What are spines? These are basically leaves only, but the leaves are highly reduced. They are very, very small. Okay. And they develop into these structures known as spines. Okay. And why are they developing into spines? In order for protection of the plant from herbivores, to prevent the herbivores on feeding on them. Okay, so that's the reason they developed spines. One more reason why spines are developed is to prevent transpiration. What is transpiration? Water loss, right? And we know that opuntia, it grows in desert. Desert has less amount of water, Desert areas have less amount of water. So plants need water for survival, and the soil has very less amount of water. So they can't afford to lose water. So that's the reason the leaves are very, very small. They're highly reduced and develop into spine-like structures. Spines, okay, which are prickly, that means thorn-like structures, but they are called as spines. These are modifications of the leaves. Okay, thorns are completely different. Thorns are also modifications which help in preventing water loss or protecting the plant from herbivores, but thorns are modifications of the stem or modified axillary buds. But spines are modified leaves. Here, the leaf only becomes spines. The leaves are very, very pointed and small, okay, to prevent water loss and to prevent the herbivores from grazing them. So, it's seen in opentia and even in prickly poppy plant. Opentia common name is prickly pear, okay, and this is prickly poppy. So that's about modifications. Leaves, apart from performing photosynthesis and transpiration or exchange of gases, they also help in providing support by modifying into tendrils, or they store food by forming fleshy leaves, or leaves help in protection of the stem by by modifying themselves into scaly leaves, or leaves get modified into spines by protecting the plant from grazing or preventing excess water loss by transpiration process. Now let's understand what are insectivorous plants. I told you plants are autotrophic, but not all plants are autotrophic, right? Some plants are heterotrophic. Now, insectivorous plants, they are autotrophic. That means they can perform photosynthesis. But these plants, as the name suggests, they trap insects. They trap insects and digest these insects for nitrogen source. Is it clear? They can perform their own photosynthesis, but through photosynthesis, you get carbon dioxide, oxygen, and hydrogen source. But what about nitrogen? To get nitrogen, these plants they trap insects and digest the insects. You can see here picture plant. Okay, this is Venus flytrap. This is bladderwort, and this is sundew plant. All these are basically insectivorous plants, okay, where they trap insects, feed on the insects, and get the nitrogen source. Now, why am I talking about these insectivorous plants? Even in insectivorous plants, the leaves get modified into different structures. Now, let me explain you the modifications of leaf in insectivorous plants using using three examples. The first is, let's understand pitcher plant. Can you see here? This is a pitcher plant. Now, whatever structure you see, this is called as a pitcher. Okay, like you have a structure like this, that is called pitcher. Okay, pitcher. Now, what is this picture? It is modification of a leaf. The leaf itself gets modified to form this picture. Okay, and the leaf, this picture, if you observe, it has a lid, just like our bottles have a lid or a box has a lid. The picture has a lid. Okay, now what is this lid? Now, if you see, let's assume this is a leaf. Entire leaf gets modified into picture, and the leaf apex gets modified into lid. Okay, and we know there is petiole right to the leaf. This petiole actually gets plays a role in synthesizing food and forming this tendril like structure or a wire like structure so that the pictures can hang. Are you understanding from the leaf? Is it clear? So the petioles they act as normal leaves performing photosynthesis, while the actual leaf gets modified into pitcher and the leaf apex forms the lid. Now what happens is, if there is a fly or any insect that lands on the picture, immediately the lid closes. And inside this picture you have certain digestive juices. What do these juices do? They completely digest the insect and kill the insect, and the plant utilizes the nitrogen source from the insect. Is it clear? So inside the picture, if you see inside the picture has hair-like structures and some juices. These juices are highly sticky. Why are they sticky? When the insect lands, the insect shouldn't fly away, right? So that's the reason the juices are highly sticky, making the insect get stuck to the pitcher. Okay, and once the insect lands and it gets stuck to the pitcher, the lid closes, not allowing, thus preventing its escape. And then the insect gets digested completely by the juices, and plants get its nitrogen source. Here, what you need to understand is the pitcher is nothing but a leaf that got modified into this kind of structure, and the lid is also a part of the leaf. And the normal leaf that you find is basically the petiole, which performs photosynthesis. Clear? And why are they called insectivorous plants? Because in these plants, insects are trapped to get nitrogen source, where digestive juices are secreted by the plant to digest the insect and get nitrogen. Okay. Next example is Venus flytrap. This is another insectivorous plant. Can you see the plant here? In this plant, let's assume if this is the leaf. Okay. This is the leaf blade or lamina. Yes or no? Now this leaf blade is divided into two portions in these plants. Okay. Like this, into two portions. And the midrib acts like a hinge, and the edges are modified into hair like structures. Can you see there are spines or hairs at the margins of the leaf or the edges of the leaf? And what is this? This is a single leaf which got divided into two. It's not completely divided, it is attached. Okay, this is one leaf, like the leaf blade is divided into two parts. And you can see this is the midrib, which acts like a hinge, which acts like the connecting structure between the two, just like a book. You know, an open book, when you open a book, right? Sorry, you have the book like this, right? Here, assume this is your midrib. Okay, and this is one portion of the leaf blade, this is other portion of the leaf blade. Actually it is one leaf only, that one leaf only in the midrib region. Here this is acting as one portion, this is acting as the other portion, and the edges have hair like structure. And this is Venus flytrap, just like a book closes. In the same way, here also the leaves close and trap insects. If a leaf lands on the structure, immediately the leaves close. Okay, just like a book can be closed, in the same way here also the leaves, they close. Okay, and trap the insect. Try to understand, the edges of the leaves have pointed hair here. Leaf blade is divided into two parts, and the midrib acts like a connecting structure. Okay, this is the hinge. Okay, in Venus flytrap. Again, the same mechanism. Inside, you have digestive juices here. When insect lands, it gets trapped, and the insect is digested to get the nitrogen source. Okay. Next is bladder wart.